All right, so we are going to go over statements A through K and identify them as true or false. And if they're false, we are going to edit them so that they become true. All right, number 1, the cellular immune response is mediated by B cells. Is that true or false? Good, Nathan, it is false. Good, good, good. So in order to make A correct, that would need to say the humoral immune response is mediated by B cells. Or we could say the cellular immune response is mediated by T cells. Good job. Okay, number B. No, number B, my bad. Can you tell how crazy today has been? All right, letter B. T helper cells, also called helper T cells. The T can come before or after the word helper. T helper cells recognize intact pathogen. Good, Maya, that is false. Good. So B cells recognize intact pathogen. Any type of T cell, whether it's a helper T cell or a cytotoxic T cell, they recognize the presented pathogen. So we can call that an antigenic peptide, antigenic fragment, digested pathogen, something that essentially indicates the pathogen has been phagocytosed, including the digestion part. Okay, so the pathogen is no longer intact if a T cell is recognizing it. Is the digested pathogen, or I could say the antigenic fragment, is that displayed with MHC1 or MHC2? Good. Antigenic fragment is with MHC2 because remember, antigen presenting cells are going to be what's presenting the antigen, and they use MHC2. Awesome. Okay. The recognition of antigens by a T cell requires that an antigen presenting cell first process them. True or false? Good. That is going to be true because processing them means, you know, catching them and phagocytosing them, right? They have to digest it before they can present it. So good job. All right, D. Cytotoxic T cells aid in both the humoral and cellular immune responses. So we kind of already addressed this one with A. Good. T cells are only associated, well, I guess that's not true. T cells, their main role is in cellular immune response, but which cell does technically help activate B cells? Exactly, Maya, one step ahead of me. The helper T cells do play a role both, okay? So to make this statement true, we would need that to say T helper cells, because notice here it says aid in both. Okay, but T helper cells, they can have other roles in the cellular immune response that we didn't really talk about, but mainly cytotoxic T cells are only involved in cellular immune responses. Remember, they're causing infected cells to die. T helper cells, though, are going to help activate B cells, or I should say further activate B cells. So remember, B cells kind of they have like two activations. B cells activate themselves when they recognize and digest their intact pathogen, and then they get activated again by these T helper cells. And it's the second activation that's going to help or allow them to divide and differentiate. Okay, E. Immunoglobulin A offers protection to a fetus because it crossed the placenta. That one's going to be false. IgA is through secretions. So IgA protects a newborn, assuming that it is breastfed. All right, IgG is going to be the one that can cross the placenta. All right, letter F. T cells release antibodies, whereas B cells release cytokines. True or false? Good. It's flipped. Yeah. For reviews, I like to throw in a lot of falses to get y'all to tell me what will make it correct. I try not to go too heavy one way or the other on exams. Okay, so to make that true, we just flip it. B cells release antibodies, whereas T cells release cytokines. All right, so T cells, like B cells, are specific for a particular antigen. That is going to be true. Good, because they are a part of our specific immune response. And remember, y'all, there's like a billion different ways I can refer to our specific immune response. It can be called the specific response, the acquired response, the adaptive response. Okay, so maybe not a million, maybe just 3, but still multiple. With our non-specific, it can also be called our innate immunity. Innate and non-response or non-specific go hand in hand. All right, next up. Natural killer cells and cytotoxic T cells release lysozyme to destroy infected cells. Good. What do they release? Does anybody remember? Words that this certain word just destroys me each time I have to say it. Maya. Yes, good. Perforins. I don't know. My mom is from Boston, so I learned to speak from her. And so for a long time, I couldn't say my R's. It was church or caw, like pop the cot, right? Like Bostonians did. But I'm in Texas. And so my classmates made fun of me when I had to read out loud. And so I taught myself to say my R's more like a Texan. But when there's double R's like that, perforin, I get all stumbled. So perforin, that's just what I'm sticking with. Good. Perforin and granzyme. Okay, so lysozyme is what you find in things like sweat and tears and saliva, and it is going to break down the cell wall of bacteria. But what our NK cells and our cytotoxic T cells release are the perforin, which is going to essentially punch a hole in the infected cell, and then that's going to allow granzyme to get in and other proteases that are going to go and chop up all the proteins and wreak havoc on the cell, causing it to implode. Okay. Dendritic cells only have MHC2 on their surface. Good. What do they have? Yes, they have both. MHC1 because they are a self cell. They belong to us. And they have MHC2 because they're special cells. They're antigen presenting cells. Good job. All right. The complement system is part of our third line of defense. Good. Well, how can we modify that? Good. Second line of defense. Wonderful. All right. What are we at now? Oh, last one. The classical pathway of the complement system begins when C2 and C4 are cleaved. What do we think of that? Good. That one is false. That one is false. The classical pathway begins with C1 being cleaved. Yes, so remember, you need to know the 3 pathways, where they begin in terms of which complement protein gets cleaved or activated first. And then you need to know what activates each one. So the classical pathway is activated by antigen antibody interactions. We call it an antigen antibody complex. And it starts at C1. The lectin pathway is activated by the detection of mannose, which is a sugar only on bacteria, and it is going to be what starts with C2 and C4. And then the alternative pathway detects just other antigens in a cell wall, and that starts straight at C3. All right. Any questions about these or other immune response type of questions? I've thought about it, but I think half the class would love me and half the class would hate me. All right. I have a Kahoot ready. Oh, was there only one true statement? Probably. Let me see. Does anybody have it documented? I don't have an answer key in front of me. I have to reread them all. There were two. Which ones were they? Oh, G, right? G was true. And then C was true. Ah, there we go. Thank you, Maya. C and G. Okay. I have a Kahoot that we're going to play next, and I'm going to warn y'all. I made it about 30 minutes before, and so I'm trying to read it as I talk to y'all. Multitasking, not my strong suit. I'm afraid I might have an incorrect answer in here, just my paranoia. I don't think so, but it's possible. So I'm kind of quickly trying to check them while I talk. All right, any other questions, comments, or concerns? Ooh, that's a typo. Okay, then let me go ahead and save this Kahoot, and I'm going to put it up on the screen, and we will get playing. All right, changes saved. Good. Let's play. All right, so this one is going to focus on microbial mechanisms of pathogenicity and like epidemiology and vaccines. And remember, this is recorded and everybody's going to get to see it. So if you want to use a name that people aren't going to recognize, feel free to do so. Hey, good. Pumped is like a positive word. Normally there's only like soul crushing terms on here. Oh, that's cool. I don't even know how y'all do this. John Smith, the most vague name out there. I like it. Back at Blinn, when I taught there, all of us micro teachers, we had like these dramatic nicknames because towards the end of the semester, students always like made us feel like we were crushing their hopes and dreams. And so like I was like the Dream Slayer, and the other one was like Soul Crusher, and it was funny. Because people always did pretty darn well in our classes. They would just get really dramatic at the end of the semester. My life is in your hands, Chelsea. I'm like, no, it's not. I just write the test. Y'all study. Spook. I'm so excited for Halloween. All right, let's see. 1, 2, 3, 4, 5, 6, 7 out of 11. Or, well, I'm 11, so 10. That's pretty good. I'm going to wait 15 more seconds. Can't do. Okay, no problem. All right, then we're going to go ahead and start. Go ahead and try answering in your brain. All right. All right, so key word here are viruses. And a second key word is going to be an evasion strategy, because remember with this chapter, we talked a lot about virulence factors, which are strategies and molecules used by the pathogen to essentially cause harm to the host, to weaken it. But then there are also strategies that they use to try to evade the immune response, so they can kind of get out of town. So exoenzymes are going to be more of a virulence factor that they're often using to kind of be stealthy. But really, there are some exoenzymes that technically can help them evade, but exoenzymes are going to be a bacterial strategy. So we remember that viruses contain an envelope, so that's a camouflage. Leukocytopenia is going to be where some viruses actually target like T cells and other immune cells so that they can't activate B cells to make antibodies. Antigenic variation is going to be where they change up the structures on their surface. And so that's commonly used by multiple types of pathogens. And then, which one did I skip? Oh, I think that's all of them. All right, so make sure we review our evasion strategies as separating bacteria from viruses. Good. I wrote this one a certain way, and I'm kind of curious how y'all interpreted it. So pathogens always leave the body the same way they got in. So the reason it's false is because I was referring to like body parts, right? So if I eat contaminated food, it gets in through my mouth, but it comes out somewhere different, right? So that's why it's false. But normally, most pathogens enter and exit through the same body system. So if I had said, when I eat food, that's my digestive system, right? And exiting food, part of the digestive process. So, yeah, exactly. So there are some where it's technically not leaving the body the same way it got in, because remember, there's also parenteral methods of getting in, right? You got bit by a rabid animal. Well, it's not going to spread, right, by being bit by a rabid animal to the next person. So most of the time, the portal of entry is similar to the portal of exit with respect to body systems, okay? All right, so definitely remember that you'll need to be able to compare and contrast endotoxins and exotoxins. I mean, it could be a question like this, but essentially you have to know the features of both. So gram-positive tends to produce exotoxins because endotoxins are built into the gram-negative cell wall. LPS is the endotoxin. So all gram-negatives have them all the time. So gram-positives tend to do them, but I believe in class I said that technically there might be a couple oddball gram-positives that produce them, but normally it's going to be gram-negative. Okay, you can't say always in any science class because there's always exceptions to the rule. So the best answer is that they stimulate a fever. And remember, exotoxins cannot stimulate a fever because they're proteins, and proteins would denature. And so essentially, exotoxins and endotoxins are opposite of each other. One's going to stimulate a fever, the other cannot. One is made primarily by gram-positive, one is made primarily by gram-negative. So with B, and I know nobody chose this one, but it says their mechanisms are specific to a disease. So that would be for exotoxins. So therefore, endotoxins, their mechanisms are all the same. They all produce fever and aches and pains and fatigue. And then they stimulate the production of antitoxins. Antitoxins are going to be antibodies that specifically go after a toxin. And yes, when we're talking about endo and exotoxins, we're talking about bacteria. Okay, next question. Hey, good job. So remember, a lot of times the name kind of gives things away. Okay, so catalase we've talked a lot about, that's for breaking down peroxide. Hemo and lysine. So lysine means to lyse. Hemo, right? Heme, blood. Okay, so these lyse red blood cells. Coagulase actually causes clumping and clotting. And so that's how some pathogens will try to hide from phagocytes, because with the clotting mechanisms, it can essentially hide behind those clots. And so the phagocyte can't detect it. And then leukocidins, I think I spelled that wrong. Yeah, the Y should be an I. Leukocidins are produced in order to destroy certain white blood cells. So leuco is like leukocyte, right, which is a white blood cell, which is an immune cell. All right, good job, y'all. Hey, good job. Good job. Biofilms, organisms with capsules, which are bacteria, have that sugary, sticky layer that is going to be able to start and initiate a biofilm. Good job. Keep hearing sounds and I don't know where they're coming from. Okay, yeah, so I pick up a Kleenex off the desk and then I promptly pick my nose, I get sick. So that is going to be fomite transmission. So it was brought to my attention that in the lecture notes, it says that fomite transmission is a type of vehicle transmission, and that is a no-go. So if you've read the lecture notes, please don't be confused by that. Fomite is through an inanimate object. Vehicle transmission is through like a non-living kind of an environment, food, soil, water. So just please make note that they are separate things. All right, so good job, y'all. All right, good job. So remember, horizontal transmission is just direct body contact. Kissing, sex, shaking hands, things like that are going to be transmitted by horizontal transmission. Okay. What on earth are these noises? Oh, I see. Okay, never mind. I thought I was missing something. All right. Chronic carriers. What is the reservoir if you're a chronic carrier? Man, y'all are on fire. I'm trying not to have too high of expectations for this test, only because yesterday y'all told me not to have too high of expectations, but y'all are doing awesome. All right, so this is kind of where I thought the answers would lie. So remember, contamination is just when something foreign has gotten on you, right? So Emmy is my niece, and this is a true story. We're trying to share ice cream, and I had many spoons for this child so she could use it and throw it away because she was only 2. But she decided she needed my spoon, and I found out too late that we were sharing spoons on accident. She thought it was hilarious. I didn't when I got strep throat. Anyways, so for when I put the original contaminated spoon in my mouth, right? I was contaminated. They hadn't started replicating or anything, but a foreign pathogen got into my mouth from that contaminated spoon. But then, starting about an hour later, as they started to replicate, then I have an infection. It's not until your actual health is altered that we say it's a disease. Okay, because I woke up with my throat hurting, that is a deviation from what is quote unquote normal. And so, that's why we would say I'm at a diseased state because my body has deviated from what is normal for it. Yeah, I have lots of great. She's always on my tests because Emmy loves to just sneeze in my face or cough in my face. At her jujitsu tournament, she was talking to me and then and I was like, oh, come on. Never fails. All right, so let me talk about this question first, and then I will get to the question in the chat. So the lower the LD50, the more virulent a pathogen is, because remember with LD50, LD is thinking about how lethal something is. And so the lower the number, the less it takes to kill you. Okay, so that means if it has a really low LD50, it's a super strong, super potent pathogen. So with infectious dose, it's the same concept. The lower the ID50, the easier it is for something to cause an infection and normally cause a diseased state. So remember that when it comes to using the term pathogenic and virulent, we should not use them interchangeably. Pathogenic is more, it is or it isn't. Is it a pathogen or is it not? It's not really a measure of how dangerous or infectious something is. Okay, that's when we use the term virulent. Virulent is along a spectrum. Ebola is more virulent than COVID or whatever. Okay, I'm just kind of making this up as we go. But you can compare things based on how virulent they are. So both Ebola and COVID are pathogenic, because either you are or you aren't. You can cause disease or you cannot. But then it's how virulent are you? That's when we start comparing infectious doses or lethal doses. And Tara, Streptococcus, it's not that it has a low LD50, it's just how many can get onto a spoon, like in my example, and I had repeated exposure. So in that example, it's not that it had just a few number of cells, it's just that there's a ton of them on one spoon and I had repeated exposure. But I honestly don't know the exact ID50 of Streptococcus off the top of my head. It is pretty virulent. As we all know, strep throat and things are pretty common. But that's also because a lot of times you can get it just like as we've seen, like with respiratory droplets and things like that. When saliva and things like that are involved, it's just easy to get a lot at one time. Okay, next question. All right, let's see here. What term is used to indicate a part of the body is usually sterile? Yep, axenic is going to be right. What else do we have? Flora, so that's going to be like our normal microbiota. Flora is just like another term for organisms that are around a lot. All right, let's keep on again. All right, just an example of a non-living reservoir. Good. So remember, vehicle is more like food, soil, water, air. An actual dust particle is going to be non-living. So the term fomite and vehicle actually refer to transmissions. They are not terms associated with reservoirs. So with reservoirs, it's either human, animal, which is where zoonotic comes in, or non-living. Those are our three categories of reservoirs. All right. So because the mosquito is needed for the organism, which is a plasmodium, to complete its life cycle, it is a biological vector. Okay, so remember with biological vectors, the vector is necessary for the pathogen to complete its life cycle. With a mechanical vector, that's where the pathogen is just hitching a ride on the vector, but it doesn't actually need that vector in order to survive. With biological vectors, the vector is needed for the pathogen's survival. All right, so capsules, flagella, biofilms, these are common evasion strategies that we've talked about. M protein is another one. It is actually used by Streptococcus pyogenes, and it is found on the surface of its cell wall, and it helps evade phagocytosis, much like a capsule. Because the M protein is kind of like this little hair-like projections that cover the cell wall, making it hard for phagocytes to phagocytose it. So latency is a viral term. Endotoxins are a virulence factor. And then protease production. Remember, proteases target proteins. And so it can go for things like antibodies or other key proteins within the cell. So that's more of like an exoenzyme virulence factor kind of thing. Sorry, y'all. I was trying to finish drinking too quickly and it didn't work. Awesome, good. All right, next question. All right, so remember, you need to be able to differentiate sign and symptom. And so signs are measurable. So you can measure, skin lesions. You can measure, whether or detect whether or not antibodies are present. So fever can also be measured with a thermometer. So those three were signs. Symptoms are more subjective. I'm tired, I'm achy, or I itch, right? It's all subjective. Good job. So the attenuated, that is the live pathogen, but attenuated means it has been weakened. And so it has been weakened to the point where it's not actually a threat anymore. But technically, because it's alive, there is always the chance that it could revert back. So inactivated vaccines are also called killed vaccines, but remember, we don't say vaccines are killed. I'm sorry, we don't say viruses are killed, right? Because they were never alive. So we use inactivated as well as the word killed so that we can reference viruses here. And then, so remember, both inactivated and attenuated use entire pathogen, whereas the remaining types of vaccines do not. They either use the actual antigen itself or the nucleic acids needed to form that antigen naturally. Good. It is a nucleic acid vaccine. It's an mRNA vaccine specifically, and so by putting the mRNA into the human body, our ribosomes and things will naturally make that spike protein. So then our body will still recognize it as foreign, right? Because it's never made it before, and we will develop antibodies in response to that spike protein. So there's no risk of actually getting COVID from the vaccine because all we're making is one little protein, not the entire virion. Good. So remember, with the incubation period, there is zero change to your body state. You still feel like you normally always do in a healthy state. With the prodromal period, that's where you can feel something coming on. You wake up and you're like, ooh, something's off here, right? Maybe your throat's a little scratchy, maybe it's a little harder to breathe, but you're okay. You can still go to work and class and stuff, but you feel off. So you know something's about to happen. So that's the prodromal period. When you have full-blown signs and symptoms, you are sick, probably miserable, but I'm also a big baby, so I'm miserable anytime there's deviations to my normal state. That's me, the period of illness. once you're starting to feel better, you're not 100%, you still feel off, but you're not, limited to the couch. Then you are in the period of decline. The period of convalescence is when you no longer have signs or symptoms, but you're still harboring a little bit of pathogen, but the numbers are very, very low. Good. So remember, with chronic, you want to report both incidence and prevalence. With an acute disease, you're only worried about incidence. Awesome job. So remember, the endemic is when something is never ending. So you typically see cases present in the population. And then an epidemic is going to be when there's a spike in the number of cases. We also refer to that as an outbreak. A pandemic is when it's on a global scale. And sporadic is when you rarely ever even see cases. They kind of just pop up out of the blue randomly. Yes, Maya. You raised your hand. I'm not sure if that was an accident. Okay, no problem at all. No problem. All right, so do make sure you know what each of these 4 terms means. All right, moving on. True or false? Good. So this is false. Morbidity is just a word you use when somebody has a disease. Sometimes people have multiple diseases at one time. So the more diseases associated with that person, the higher their morbidity level. But when we're talking about death specifically, that's going to be the mortality. So this is part of the vaccine FAQ. I wanted to throw in some of the questions here. Good, good. So viruses have to be cultivated in actual cells, right? So there are cell lines that long ago may have derived from an aborted fetus, but these are not present in all vaccines, okay? But remember, it's kind of hard to find somebody to sign up to grow some of these really dangerous viruses in them, right? And so sometimes, like I said, long ago, we would have used certain cell lines to grow viruses, but they're not in the vaccine themselves. Good. Yes, they do, but in very, very, very small and safe amounts. Good. Last question. Good job. And so remember, immune and like immunization and vaccination can't be used interchangeably, right? Because vaccines aren't always 100% effective. But in order to establish or for herd immunity to occur, most of the people need to be immune. And so that can either come from vaccinations or by being exposed to the disease and surviving it, of course. But remember, not all diseases, even if you survive it, they don't always confer lifelong immunity, right? We've seen with COVID, and I don't remember the exact number, but they think it's probably about less than a year that antibodies will remain after you've cleared COVID. And again, don't quote me on that exact number, but they're not thinking that it offers lifelong protection. And so vaccinations are going to be our best bet for that. Upwards of in the 90s is normally where it's going to start to happen, but pretty darn high. All right, y'all. So that is all I've got for y'all. Are there any questions that I can answer? All right. Well, then we are. Oh, hold on a second. Oh, hold on. My dogs are going bonkers. That's okay. So yeah, some exoenzymes, depending on what they are, can be an evasion strategy. When I was writing that question, I was trying to think of like actually like really getting out. So some exoenzymes are and some are not. So that probably wasn't my best question. But some do help them evade, but some also contribute to virulence. All right, any other questions, comments, concerns? Yes, Nathan.
So the natural killer cells and the cytotoxic T cells, those are both going to release the lysozyme. I mean, they're both going to release the two
yeah, perforin and granzyme.
Okay. Yeah, okay. Sorry.
No, you're fine. Anything else? All right, y'all. Have a great rest of your day, and I will talk to you later. Feel free to reach out if you have any other questions.